Hello everyone, welcome to today's live broadcast, Sensitive and Specific Multiplex Real-Time PCR Assays for assessing the abundance of extremely rare mutations associated with cancer diagnosis, prognosis, and therapy. I'm Christina Jewell of LabRoots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Before we begin, just a couple of important announcements. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A lower left. I would now like to introduce today's speaker, Dr. Fred Kramer. Fred Russell Kramer is professor of microbiology, biochemistry, and molecular genetics at the New Jersey Medical School at Rutgers University and has been a principal investigator at the Public Health Research Institute for the past 30 years. He graduated from the University of Michigan in 1964 and received his doctorate from the Rockefeller University in 1969. He was on the faculty of the Department of Genetics and Development at Columbia University College of Physicians and Surgeons for 17 years and was a research professor and adjunct professor in the Department of Microbiology at New York University School of Medicine for 27 years. I will now hand over to Dr. Kramer for his presentation. Dr. Kramer?
Okay, I hope you all can hear me. Let me put on my slides. Let me make sure. Okay, very good. Okay, what I'm going to talk about today is the work of 4 members of our laboratory, shown on the screen: Salvatore Maris, Diana Vargas, Sanjay Tyagi, and myself. And basically what I'm going to be speaking about is a way to do liquid biopsies for cancer. Now, what do I mean by a liquid biopsy? Imagine that if you go into the doctor's office for your annual checkup, and in addition to taking a blood sample to check for your cholesterol or whatever, they take a blood sample, a small blood sample, say 10 ml, and they use that blood sample to look for somatic fragments of DNA in the blood plasma, which contains somatic mutations indicative of the presence of cancer anywhere in your body. This is quite possible. It's known now that cancer cells rapidly divide and also rapidly die from apoptosis and necrosis. And as a result, those cells are chopped up by enzymes, and fragments of the genome of the cells that have died end up in your bloodstream for a short period of time. Wouldn't it be wonderful if taking a blood sample in a regular checkup could detect extremely rare somatic mutations indicative of cancer long before there's any symptoms, and it would come from anywhere in your body. But by identifying which mutations you have, you'd be able to early on treat before there are symptoms. And hopefully the idea is, if the assays are sensitive enough to detect these very rare DNA fragments, that you can successfully treat a person, which might be much more difficult if you waited until symptoms occur. I'll give you other examples of the advantage of having a very simple blood test for cancer. If you've been treated for cancer, if you have had a tumor removed or a bone marrow transplant, right now there's still a worry that you have cancer cells in your body. It's the problem of minimal residual disease. Therefore, people are automatically given very toxic drugs to treat the potential that cancer could return. Wouldn't it be much better if there was a simple blood test so sensitive that it could detect the presence of those remaining cancer cells. And not only that, because of clinical studies done with that test, you could determine at what levels it's not necessary to add any drugs. And you could follow that patient over time, periodically testing the patient. And only if the level of cancer cells goes above predetermined amount would you then treat the cancer with a toxic drug. Another example is if you're being treated with a drug, you know that cancer cells can develop mutations which generate resistance to that drug. Right now, you often have to wait until you have symptoms. That's not good. Wouldn't it be better if a patient could be followed regularly and those mutations detected long before there are any symptoms so that the drugs administered to that patient could be changed? That's personalized medicine. One last example. Some people inherit mutations which aren't cancer per se, but which are defects in repairing mutations that occur. These are the BRCA mutations. They often can allow mutations to occur that lead to cancer, breast cancer, ovarian cancer. And if you wait until symptoms come from these, it's often too late and you die. So many women have prophylactic breast removal or ovaries removed. Wouldn't it be much better if there was a test so sensitive that periodically these women could be tested for the occurrence of cancer, and early on, if it's detected, do something about it, cure it, and therefore there wouldn't be necessary to have prophylactic breast removal or ovaries removed. So the whole talk is about how you could take a simple blood test or plasma from a blood sample and test it for the presence of rare mutations. And the single biggest problem that has to be overcome is that almost all the DNA fragments in your blood, in your plasma, come from normal cells. And the only difference between those fragments from normal cells and fragments from the cancer cell is often just a single nucleotide substitution, a single nucleotide polymorphism. So how can you do a simple, low-cost test that can be administered frequently and yet selectively analyze that sample for those rare somatic mutations? And so the test that we've been working on is a polymerase chain reaction, a PCR test. And first, let me show you the problem in the first slide. Here are two PCR reactions that we ran, each initiated with 1 million DNA fragments, but one was with wild-type fragments and the other was with mutant fragments. And you can see that using a primer, which is diagrammed in the upper left-hand corner, where one nucleotide in the middle, that's the open circle, is complementary to the mutation, but not complementary to the wild type. Makes no difference. The primers bind equally well, and you can't distinguish the wild type from the mutant. What I'm going to be talking about today is we've invented a new type of primer, a primer whose functions are broken into two parts. I'm going to try and use a little pointer here, sort of. Over here on the 5' side of the primer, you see I'm indicating it with a little red line, is something that looks very much like an ordinary PCR primer. And it allows that primer to selectively bind only to the fragments of interest, to ignore all DNA fragments in the plasma that are not related to the gene of interest. And then on the other side of this primer is a very short segment, which is complementary to the region that either will contain or not contain the mutation. And because this is short, it then will make a difference between whether that foot, that little 3' end there, is able to bind to the target or not. If it matches the mutant completely, even though it might be only 7 nucleotides long, it's going to rapidly initiate the synthesis of a product. Whereas if there's a mismatch between that interrogating nucleotide, shown by the open circle, and the target sequence, that is, if it's a wild-type target sequence, you can see there's almost a 20 cycle delay before any product is made. So this type of primer, where it's been broken into two functional regions, a 5' region that selectively binds the primer only to the target of interest, and then a short 3' region that, because it's short, is able to bind only to the mutant that you want to detect. And joining the two regions is something we call a bridge, and it's unrelated to the sequence that you're trying to detect. It's just quite separate. But what we've done here is we've allowed ourselves to get a very small primer, the foot primer, to make the distinction. Had we used just a small primer by itself, it would have bound to 10,000 different genes and wouldn't have done the trick. Okay. Here is the principle of these assays, sort of illustrated. On the left, you see one of these, we call them SuperSelective primers. And if the interrogating nucleotide, which is a G in this case, matches the mutant nucleotide, a C, the foot sequence will bind, and that can initiate the synthesis of amplicons. Whereas on the right side, if there's a mismatch, the foot does not bind, and it's very unlikely to initiate the synthesis of products. Here's a closer look at what the SuperSelective primer looks like. The primer's on the top, and the DNA fragment that you want to detect is on the bottom. And you see the red nucleotides is the anchor sequence that serves the function at the 60-degree annealing temperature of bringing the primer to the targets of interest. And then on the right side, the green nucleotides show the foot sequence containing the interrogating nucleotide, which, if it's perfectly complementary to the foot target containing the mutant characteristic of cancer, amplicon will begin to be made. Synthesis will occur. And you'll notice that the blue nucleotides of the primer, the bridge sequence, is not complementary to an intervening sequence in the target that separates the anchor target from the foot target. And there's a little code in the lower left there. This is a primer that selectively amplifies the cancer mutation, BRAF V600E. And you'll see the code 2414/14511. That means the anchor is 24 nucleotides long. The bridge sequence is 14 nucleotides long. The intervening sequence is also 14 nucleotides long. And the interrogating nucleotide is at the next to the last position of the 7 nucleotide long foot. That's why it's called 511. Here's a closer look. There's that same primer for BRAF V600E. And at the bottom, you can see the PCR conditions are absolutely normal, ordinary. The only difference here is we substituted a SuperSelective primer for the usual forward primer. And there's a regular conventional reverse primer, and conditions are perfectly normal. One of the special and most wonderful things about this approach to selective PCR is that the selection only occurs when the SuperSelective primer binds to a minus target strand. We arbitrarily call it minus. And the foot binds and gets extended. So that the upper diagram shows the SuperSelective primer being extended because the foot matches the foot target. However, in the next thermal cycle of the PCR, the conventional reverse primer copies the whole amplicon generated in the top diagram, making a complete copy of the entire SuperSelective primer sequence. That is, the bridge sequence is now copied, and a complement of the bridge sequence ends up in the new minus strand that's made. So that in the bottom diagram, in the third cycle, that minus strand now serves as a quite ordinary template for the SuperSelective primer. And the SuperSelective primer acts as an ordinary primer. So the moment a selection event occurs on a DNA fragment that's in the original sample, then the PCR is efficient and normal. And that's a beautiful thing. So that's what it is, that selection is only each cycle on any initial DNA fragments that are present. Here's the result of using a superselective primer. And I think this is a key slide. So we're looking here for this BRAF mutation V600E. And there are 8 different samples that were run. If there's no DNA present at all, the red line shows you that nothing is made. If we started a reaction with 1 million wild-type templates, that's the green line, you see you do get amplicons made, but they don't appear until about the 50th thermal cycle. The initiation on the million wild types is incredibly low probability. And the blue reactions all contain 1 million wild types, but different numbers of mutants. And you see that depending on how many mutants are present in the sample, that determines when there are enough amplicons to see. And the fewer initial mutant templates that are present, the later those amplicons arise. But yet, even 10 mutant molecules in the presence of 1 million wild types occurs earlier in the reaction than pure wild types. The machines, you can use any PCR machine to do these reactions. In this particular case, we're using cyber green to light up the amplicons. The instruments automatically, that's that dotted line, determine a level in which synthesis has risen above background. And when the reaction has achieved a thermal cycle at which there is a product, amplicons above background, that's noted by the instruments and can be plotted in the following manner. There's a relationship between the threshold cycle, where the amplicon products above background occur, that's inversely linearly proportional to the logarithm of the number of target molecules present originally in the sample. So you see, you get this beautiful straight line. And this relationship between threshold cycle, inverse relationship between threshold cycle and logarithm of the number of targets was introduced by our laboratory in 1989 in articles in Nature and in Clinical Chemistry. And it's the basis of real-time quantitative PCR assays. One more thing to plot here is what is the threshold cycle of the pure wild type. And here it is plotted as a dotted green line. You see that the 10 molecules of target, even in the presence of 1 million wild types, can be distinguished from 1 million molecules of wild type. That's how sensitive and selective are superselective primers. However, nothing works perfectly. When we repeat this experiment, but this time we use a different mutation as a target, the EGFR L858R mutation. Now, what distinguishes this mutation from the BRAF V600E? The BRAF V600E mutation occurred in an AT-rich region, whereas the EGFR L858 mutation occurs in a GC-rich region. And you see, when you do exactly the same experiment as what was done with the BRAF, now the 10 target molecules can't really be well distinguished from the pure wild type alone. And so it's clear that the nature of the target, the strength of the foot hybrid that's formed, is going to affect the selectivity. And so we looked into this in more detail. For instance, if we lengthen the foot sequence, things get even worse. You see now there's really no difference between 100 target molecules in the presence of 1 million wild types. On the other hand, if we shorten that foot sequence, making a less strong foot hybrid, then you return to that full linearity. So making the strength of the hybrid that's formed is going to affect the selectivity. That's our first discovery. Not only that, we also looked at the position in the foot of the interrogating nucleotide. And we just compared reactions with 1 million wild types to reactions with 1 million mutants. And what you can see is having the interrogating nucleotide at the 3' end or at the next to the 3' end, where we assume that if the penultimate base pair doesn't form, at 60 degrees, the terminal base pair will also not form. You get good discrimination. But as you move that interrogating nucleotide away from the 3' end of the primer, discrimination is not as good. We also investigated the size of the bubble formed by the bridge sequence and the intervening sequence. So here are experiments done with different bubbles of different circumference. 20 nucleotide bubble, a 28 nucleotide bubble, and 36 nucleotide bubble. And again, the bigger the bubble, the better the discrimination. And what does this mean? Again, I want to emphasize the bigger the bubble. When these little foot hybrids fall apart, you'd like them to go back together again. And there's an entropy factor. The bigger the bubble, the less likely it is that the foot and the foot target will come together. So in effect, you have weakened the free energy formation of that foot hybrid, but that leads to better discrimination. Okay, one other thing we looked into in the bubble is, what if it's not a symmetrical bubble? What if the bridge sequence is bigger than the intervening sequence or vice versa? And basically, I won't dwell on this, it doesn't matter. All that matters is the size of the bubble, but the symmetry of the bubble can be altered, and that's going to be very helpful in the design of the experiments that I'll be describing. Okay, by taking this EGFR L858R mutation, which didn't give the best of results because it was a GC-rich target, we now repeated that experiment using a shorter foot and a non-symmetric, slightly larger bubble. We made a bigger bridge. And then you see the results are beautiful, that you're able to discriminate even 10 mutant molecules in the presence of 1 million wild type DNA templates. So we're going to come back to this, but a key factor in designing this new way of doing extremely selective, extremely sensitive PCRs is that you have to take into account the strength of the foot hybrid that forms. It's going to affect the outcome, and we'll get to that. Now, we did these experiments with linearized plasmids, the ones that I showed you so far. In this way, it sort of mimicked what you would find in a liquid biopsy sample. But they only had a small segment of the gene of interest. The question is, what happens if you use all genomic DNA? And we can get genomic DNA from cell lines. And we had, for the EGFR L858R mutation, we had a cell line containing that mutation and another cell line that was wild type. And for this, a liquid biopsy is likely to have DNA from something like 10,000 cells. So we used DNA from 10,000 wild type cells. And on the right side, the result is what you got with that green reaction. But if you mixed in with 10,000 wild type DNA from 10,000 wild type cells, the DNA from 10,000 mutant cells, and by the way, these were fragmented with restriction enzyme, you got a beautiful result. It doesn't matter if it's the whole human genome that's in the sample, because the anchor sequence of the SuperSelective primers assures that the only fragments that are paid attention to are those that are complementary to that anchor sequence. One other thing, we wanted to see if the threshold cycle is really reflecting the number of DNA fragments that there are in the sample. So working with a company in Portland called Molecular MD, they used DNA from cell lines to and they intermixed DNA from EGFR, a cell line containing the EGFR mutation T790M, with about wild type DNA from about 13,000 wild type cells. And they ran a digital PCR assay to count in a set volume the amount of the number of molecules of targets that were in there. And they sent those samples to us, but didn't tell us how much was in each sample. And we ran them with our PCR assay and determined the threshold cycle for each using the same volume. And when after we ran the reactions, they sent us the results, we compared them. You can see that there's a beautiful correlation between the threshold cycle that we measured and the number of targets in the sample as determined by digital PCR. So how does this work? Why does this work? One of the key things is that the foot hybrid of the mutant target is going to be 7 long, let's say, and the foot hybrid with the wild type will be perhaps 5 nucleotides long, less strong. So we did thermodynamic calculations to see if this could account for this 20-cycle difference in the appearance of the amplicons. And that's a million-fold difference. And the answer is thermodynamics does not account for such a big difference. Something else is going on. And this is why I put emphasis on the nature of the foot hybrid itself, because stronger hybrids like the EGFR L858 made it a little harder to discriminate than weaker hybrids, such as the original BRAF V600E, which were AT-rich. So the nature of that foot hybrid is also affecting the outcome. And we now believe we understand this. It turns out that the anchor hybrid, 24 nucleotides long, is so strong that it persists throughout once formed, it persists throughout the whole annealing stage of the PCR. 20 seconds, it probably exists persists for a minute or so. But these little bitty hybrids exist for less, the foot hybrids exist for less than 1 second, maybe only a few hundred milliseconds. And it turns out that the mismatched hybrids, the hybrid of the foot with the wild type, that is so short, it only exists for a few milliseconds. So there's a big difference in the persistence time of these hybrids. I mean, it's at equilibrium, they're falling apart, going back together. But basically, when the hybrid is formed, it has to encounter a DNA polymerase and get the formation of the amplicon started. And the 7 long fully complementary hybrids with the mutant, they're able to pretty much encounter a DNA polymerase. But the mismatched hybrids of the wild type are so short and last for such a little time, they don't have time to encounter a DNA polymerase. They fall apart before that. So let me sort of summarize this. In the past, people have tried to get highly selective assays by making a good, strong mutant hybrid, primer that makes a strong hybrid, and make a weaker, a much less strong mismatched hybrid with the wild type. That worked, but not great. What we've done is we've made both the perfectly matched hybrid and the mismatched hybrid very, very weak. But because we've made them both weak, the mismatched wild type hybrid is so weak it hardly exists. It falls apart. So there's this second factor by making everything weak. And that is that the mismatched ones don't even last long enough to get a DNA polymerase. And so the combination of thermodynamics and this difference in the short lives of the foot hybrids, that accounts for the incredible selectivity of our primers. However, just all of these things I've shown you are monoplex assays looking for just one mutation. Practical assays that are going to look at liquid biopsy samples need to look at multiple mutations. Not only that, many of these mutations, although they occur in different cells, will occur be located in the same or adjacent codons. That's the case for the KRAS mutations and the BRAF mutations and the EGFR mutations, all of which are relevant to cancer diagnosis, prognosis, and therapy. So we need to have a multiplex assay, and we need to have a multiplex assay that overcomes the problem that there'll be different amplicons that are virtually identical to each other. So here on the slide, we're showing the 5 problems that need to be overcome. We were using cyber green for the monoplex assays, but in multiplex assay, you need to use differently colored probes that can light up each of the different amplicons that would be generated if that mutant target is present. Ordinarily, if one had genes in totally different chromosomes, there'd be no problem finding a probe that could uniquely bind to each of the different amplicons. But since these amplicons are virtually identical to each other, the same anchor sequences, virtually the same rest of the amplicon, you can't use a fluorescent probe that goes into the middle of the amplicon. So that has to be solved. The other thing is with primers sharing the same anchor sequence, say for the KRAS mutations, how do you make sure that only the right primer out of the set of primers that go to the same gene will go on an amplicon? If an amplicon has one particular mutant out of 4, and there are 4 different primers, you've got to make sure that only the correct primer goes to the amplicon. That's problem number 2. 3, if there are 2 mutants are present that occur in the same gene, the same codon, if one is more abundant than the other, you know that during an amplification, you keep on making plus and minus strands until finally there are so many that they start to form double strands. That's why PCR reactions go from being exponential to quasi-linear, because it's hard for the primers to get in there into the double strands. It goes quasi-linear. But if you have now a combination of amplicons, one from an abundant mutant and another from a much rarer mutant, once the amplicons from the more abundant mutant start to form double strands, the less abundant mutant will get caught up as heteroduplexes in those double strands, and they will not be able to undergo continued exponential amplification. That has to be solved. And finally, we've seen that each different target having a different sequence has a different propensity to get initiated. So the threshold values vary. So we have to have some way to fine-tune all of the primers that are put into a multiplex reaction so that the same threshold cycle from any of those primers will represent the same number of targets in the original sample. And the last problem is that we don't want to have to determine the amount of DNA that we put into a sample. And in fact, if you take a plasma sample, a blood sample from the same patient a few hours apart, and you take a number of samples, you'll find that there are different amounts of DNA fragments in each sample. It varies quite a bit from hour to hour. So we want to add into our multiplex reactions the amplification of a reference gene whose threshold cycle will tell you how much DNA is in there. And you'll see that that's very important. So let me quickly go through the various things that we've done. First, in order to detect the different amplicons, we're using molecular beacons, which is a fluorescent probe developed in our laboratory. You can see from the picture that they're little hairpin-shaped probes, where the probe sequence, the part that matches the target, is complementary to the target, is in the loop of the hairpin, and that probe sequence is embedded within 2 arm sequences that are unrelated to the target sequence, but they're complementary to each other, so they form this little hairpin stem. And on either end of that probe is a fluorophore and a quencher. And the molecular beacon floating around in solution is completely dark. It's not fluorescent. And the reason is that fluorophores do not have side groups that wiggle, waggle, or rotate. So that if they are stimulated, if they absorb energy from light, they don't have side groups that can disperse that energy through the motion of side groups as heat. So instead they fluoresce. But the quencher molecules that we use do have side groups that wiggle, waggle, and rotate. And when the quencher and the fluorophore are close to each other, bonds are formed between them. Hydrogen bonds, pi bonds, whatever. And so when the fluorophore in the floating around molecular beacon absorbs energy from the stimulating light, it passes across those bonds to the quencher, whose side groups release that energy as heat, and there is no fluorescence. It's only when the molecular beacon binds to its target that the rigidity of the probe-target hybrid forces those arms apart, then breaking the bonds between the fluorophore and the quencher. And then when the fluorophores absorb energy, they only can release it as fluorescent light of a pre-chosen color. And you can use a variety of different colored fluorophores in multiplex assay using a variety of different molecular beacons. So here's what we did. I think I will get the little marker here. Let me see. Yes. So in addition to the anchor sequence and the bridge sequence and the foot sequence, we add to each superselective primer a 5' tag sequence. And this isn't going to affect the selectivity or initiation of synthesis. And you can see that once the amplicon begins to be made, you have normal exponential amplification, producing both plus and minus strands. And the minus strands contain a complement of the 5' tag sequence of the primer. However, we also, when we set up these reactions, reduce the concentration of the superselective primer compared to the concentration of the common reverse primer. So we might have 60 nanomolar superselective primer, but 500 or 1,000 nanomolar of reverse primer. So what happens is exponential amplification runs out when all the superselective primers for that target are used up. But the reverse primer, which is more abundant, keeps on making products. So you get an excess of minus strands, and that's shown in the lower diagram. And we have in the reaction a molecular beacon whose probe sequence is complementary to the complement of the 5' tag sequence on the primer. So the result is that once exponential amplification has run out, you get a linear synthesis of minus amplicons, which are lit up by a molecular beacon whose probe is complementary to that 5' tag. And in multiplex assays, we're going to use different superselective primers, each containing a different tag sequence, and we're going to have different molecular beacons, each containing a differently colored fluorophore, and each of them only bind to their own tag sequence. Here's an example just of a monoplex reaction for an EGFR G719S mutation using a molecular beacon. We used 40,000 wild-type fragments and different numbers of mutant fragments, going to as little as 4 mutant fragments, and you see what beautiful results you get using a molecular beacon to light up the amplicons. Now, here's an example of a multiplex, a duplex reaction. So you see there are 2 different molecular beacons there. One for the 5' tag sequence of a V600R superselective primer, and the other one, the red one, for complementary to the, or not complementary, actually the probe sequence is identical to the 5' tag sequence. The probe sequence in the beacon will be complementary to the complement of the 5' tag sequence. Now, there's one other aspect that we have to worry about. When you have 2 or more different primers for virtually the same gene, you have this, how are you going to assure that only the correct primer binds to the amplicon? So here, let me use this little pointer again. This whole bottom strand here, that's a minus strand to which superselective primer has to bind. Now, it's quite possible that an incorrect primer, which shares the same anchor sequence as the correct primer, will in fact bind to that minus strand. Even a molecular beacon could bind. But because each superselective primer has its own unique bridge sequence chosen by us, only the correct primer will bind to the complement of that bridge sequence that's in the amplicon. So this assures that no matter what happens, each amplicon is only copied by its own correct primer. Another problem that occurs, in this case, we purposely ran reactions in which, in addition to 10,000 wild-type, BRAF wild-type templates, we put in every reaction 1,000 copies of BRAF V600E mutant. But in each reaction, we put in different numbers of V600R mutation. And what we realized is that the V600Es will all sort of reach their threshold cycle, somewhere around 35 cycles here. But you'll notice that the BRAF V600Es, instead of having separate threshold cycles, they get caught up in a heteroduplex with the V600E. And so you can't really get proper threshold cycles for V600R. This demonstration was done using equal concentrations of the SuperSelective primer and the reverse primer. However, when we go to using only 60 nanomolars of each SuperSelective primer and having much more of the reverse primer, then you don't have the heteroduplex problem. There's less plus strands of the more abundant primer to get tangled up with the rare minus strands of the V600R primer, and you get a beautiful separation, look on the left, of the V600R primer. And there's something else that helps. Here is a picture that shows a red amplicon from a plus strand from a mutation whose SuperSelective primer has been used up. And it's bound to a minus strand of a very rare mutation that is still undergoing exponential amplification. And what you can see is that it's not a perfect duplex. In fact, and I'll use the pointer again, the bridge sequence of the plus strand of the more abundant amplicon is not complementary to the complement of the different bridge sequence of the less abundant amplicon. As a result, you get an accessible bubble, and it's right here in this minus strand complement to the bridge sequence that the SuperSelective primer for that rarer target can get in there, bind, and generate amplicons. And that's why the heteroduplex problem is solved. Further, since we know that different target sequences having different GC contents, what have you, different sequences, have a differential efficiency, and you can see that in the left-hand panel is a duplex reaction run with a V600R and a V600E primer. And everything's the same in terms of quantities, but the threshold values of the red one, which is the V600E, came later. The V600E is very AT-rich, and the threshold cycles came later. So we are able to fine-tune the situation by altering the size and even somewhat the sequence of the bridge sequence that's in the SuperSelective primer for V600E. So you see this one that didn't work so well in the left-hand panel. It had a 15/9 bridge sequence, and the threshold cycles came too late. But then when we redid exactly the same experiment using a BRAF V600E SuperSelective primer with a 10/9, is shorter, its circumference is shorter by 5 nucleotides. This made the entropy, the getting back together of the foot and the foot target hybrid, more probable. And then the very same threshold cycles on the left indicate the very same number of target molecules on the x-axis in the right-hand diagram. So we're able to fine-tune all of the SuperSelective primers that we put in because our SuperSelective primers have a unique sequence chosen by us whose identity and size we can vary. Now, the last thing is we wanted to have a reference gene also made. Now, here's a triplex reaction. The reference gene happens to be a cancer target that we have around, but we're using the wild type. So it could be beta-actin, it could be anything, but we happen to use the bottom superselective primer is for EGFR wild type. And you see there are 3 different superselective primers. There are reverse primers, a separate reverse primer for the EGFR and a common reverse primer for the BRAF. 3 different molecular beacons, each labeled with different colored fluorophores. And other than that, the superselective primer concentration is low and the reverse primers are high. It's a quite ordinary PCR. We're looking at the fluorescence at the end of the annealing stage. And we're looking at the fluorescence from 3 different colors. So I want to show you how this works. So there's 2 experiments here I'm going to show you the results of. So in these reactions, first of all, every single reaction contained 10,000 BRAF wild types, but there's no primer for the wild type. They also contained 10,000 EGFR wild types, which are going to be a reference gene target. And then in the experiment shown on this screen, in every tube there are 1,000 BRAF V600E targets, but different numbers of V600R targets. And what I want to show you, I'll use the marker again. I think this is it. Yeah, so here we're in the red. You can see that the CT value of the wild type reference gene is the same for all reactions. The CT value looked at in a different colored fluorescence is the V600Es. They have a common CT value for the 1,000 targets, but the CT values for the V600Rs varied depending upon how many were in there, all the way down to 10. If you take these results in the lower right-hand corner and plot them, you find that all of the CT values for the V600R lie on a straight line. The CT value for the pure V6 wild type, that is, if we look at the fluorescence that we get for the molecular beacon for V600R, when there are no targets in there, the CT value is up here. It's this 55 or so. And all of the ones that contain mutations right down to 10 lie on this straight line. But here's the other thing, because we fine-tuned everything, all 1,000 V600Es give you the same CT value, and all of the reference genes give you the same CT value. What does this mean? Well, before I tell you what this means, here's the reverse experiment. Everything's the same, except this time we used 1,000 V600R targets in every tube and different numbers of V600E targets. And again, when you plot it, everything goes on a straight line. This tells you that you can do multiplex reactions using a different superselective primer for each target. The number of probes primers you put in depends upon the number of colors you can discern. If you don't care whether 3 or 4 mutations, which ones they are, but you only want to see if any one of them occur, you can put in 4 superselective primers, all with the same 5' tag sequence that are lit up by the same molecular beacon. And if you get a signal from any of those, that'll be satisfactory. But again, because you have a threshold cycle value for a reference wild type, that tells you how much DNA is in there. And the fact that you get amplification tells you that everything's working well, the PCR is going. And where the threshold value comes up for the reference gene, that tells you how much DNA you have. And if that threshold cycle comes up too late, it means you don't have enough DNA to look for the rare targets, that sample is not satisfactory. But if it comes up early, as will generally be the case, all you have to do is compare the threshold cycle of the reference gene to the threshold cycle of the mutant that you're going to detect, if it's there, to give you a measure of the abundance of that mutation. And that's clinically what matters. So I would say, I really have a conclusion here, that if you use fine-tuned superselective primers possessing primer-specific sequences for detection and for target selectivity. You combine that with non-symmetric primer concentrations and the use of an internal reference gene target, and this enables the routine use of PCR reactions to detect and quantitate rare mutations that occur in the same or adjacent codons in the presence of abundant wild-type sequences. This means that this approach, low-cost, simple assays done with blood samples that are easily taken frequently, can be used routinely to manage the care of individual patients. That's the promise of this. Okay. I just wanted to show you my colleagues. Here's Salvatore Maras. He's been in the lab, I don't know, 15, 20 years, something like 20, I think. And he is from the Netherlands. Here is Diana Vargas. She's from the country of Colombia. She's been in the lab 15 years. All these experiments were actually carried out by Diana. And here's my long-term laboratory colleague, Sanjay Tyagi. He's from India. We've shared a lab for 29 years. And if you would like to hear more about this approach, if you would like to try it out, if you'd like to collaborate with us, please contact me at the following email address. And we'll get in touch with you and see what we can do. And that's basically my talk, and I was wondering if there are any questions.
I'm sorry, I believe I didn't have any audio for a minute. Dr. Kramer, thank you so much for that great presentation. Now, before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit the questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Okay, Dr. Kramer, our first question is, what is the best way to use positive control for liquid biopsy PCR? What should I use? Amplicons, plasmid, or cancer tissue DNA samples?
Okay. All of the work that we've done in our laboratory has been either with plasmids or just DNA fragments, or with DNA isolated from tissue culture cells. That is basically, we're not clinicians, and we've done all of our work with model templates. I think now it's time for actual clinical samples to be used. So if you're developing an assay, you could well do it the way that we did it, which is use artificial targets to get everything fine-tuned and set up, and you're confident with it. But then one has to check it out using clinical samples, which we haven't done yet, and then ultimately you're going to make measurements on already previously characterized clinical samples. And I don't know the source. I'm not a clinician, but the thing that you'll measure with the assays is the difference between the threshold cycle of the reference gene and the threshold cycle of the mutation, should it be there. And one would want to correlate that difference, where a smaller difference means there's more mutations there with the symptoms that are present or with the previous measurements that are done by other methods. I should say one other thing, and that is that you might use a low-cost, simple, rapid PCR assay to search for mutations. If you find something positive clinically, it might be then appropriate to do a more elaborate, much more expensive next-gen sequencing to look at multiple mutations that are relevant to the situation. But I mean, what we're looking here is to get something that could be used routinely, frequently, for people who don't have cancer and for people who have cancer, to get to the problems that occur much, much earlier, and therefore it's much easier to treat the patient with better outcomes. I think that's the best that I can do. Okay. so you can regain control.
Next question, Dr. Kramer. Does your PCR primer design give advantages for digital droplet PCR?
Am I on now? Look, that's a very, very good question. It does in the following sense. These mutations are so rare, and especially if you want to look for multiplexes, that you'd have to do millions and millions of droplets in digital PCR, millions and millions of wells. Because you have to distinguish your product that gets made, it's all or none, it's endpoint in digital PCR, from wild type. But if you use super selective primers, let's say in every droplet you have one or no mutant target initially, and then that's amplified. But you might have 100 or 200 wild type targets because they're so much more abundant in that droplet. But if you use super selective primers, those wild types will easily be repressed. They won't make amplicons. So the answer there is yes, super selective primers will help in digital PCR. There's something else, and that is I've talked about molecular beacons using, say, 5 or 6 or maybe as many as 10, depending upon the instrument that you use in PCR. But our lab has actually worked on ways to label molecular beacons with 3 different colors out of, say, 7. And there are 35 different ways to choose 3 colors out of 7. And a molecular beacon for each of 35 different targets can be labeled with a unique set of 3 colors. And it's not done by putting 3 fluorophores on one molecular beacon molecule. It's by taking one molecular beacon molecule without a fluorophore, but with a quencher, divided it into 3 test tubes, and then in one test tube, say, put in a green fluorophore, another test tube add on a red fluorophore, and a third test tube add on, say, a yellow fluorophore, then mix them all together. You now have one molecular beacon with one probe sequence, but a third of them are red, a third of them are green, a third of them are yellow. So you could have 35 different unique molecular beacons for unique tag sequences, and even though you only distinguish 7 colors, in which there's only going to be 3 for any droplet, unless there's a rare chance that 2 targets have occurred in the same droplet, you can do highly multiplex digital PCR. But this requires a PCR machine that can discern, say, 7 different colors in each droplet, or it can take a spectrum and then dissociate it mathematically into the contribution of each of 7 colors. Another alternative is to use beaming, where the amplicon products and their probes are attached to little particles. Those particles are sent through a flow cytometer, and the flow cytometers can take a spectrum of each particle and dissociate them. So yeah, there are a lot of possibilities to combine this with digital PCR, but I'm still an advocate of low cost, $20 to $50, depending upon, who knows, market conditions, to run an assay with regular ordinary PCR. There must be 100,000 spectrofluorometric thermal cyclers spread throughout the world already, and assays could be made for any of them. Okay, I think that's the answer to this.
Thank you, Dr. Kramer. We have great questions coming in, but we have time for one more question. Our audience member says, interesting talk, Dr. Kramer. For the last slide you showed, I would like to confirm the CT values. From my previous work, anything more than 40 CTs is considered less than 40 CTs is considered low sample. Please clarify why your graph starts to rise starting at 40. I understand that you set the cycles up to 60. What is the state of your sample?
I don't think I fully understand. I don't think I fully understand the question. Because we're using super selective primers whose foot and foot target hybrid is very small, the probability of initiating synthesis is nowhere near 100%. It's a sort of low probability. That makes the threshold cycles come out later. However, once an amplicon is generated, then it's exponentially amplified. And of course, there are multiple targets in there, so there are multiple of any kind. And that means there are multiple chances to get started. Also, in each subsequent thermal cycle, the reverse primers copy the plus strands, making more minus strands. So there's a linear increase in the number of target strands for the superselective primers as the a reaction occurs, the PCR reaction occurs. So we find that it's highly reproducible, that the threshold cycle that you get, even though it's later than conventional PCR for the same number of targets, it reliably reflects the number of targets that were in the sample. That's why you get these beautiful plots of the threshold cycle being inversely related to the log of the number of targets that were in the sample. And not only that, I'll say this, that we wouldn't have time to go into it, that we're working on ways to assure that even if there's only one or two target molecules in a sample, that you will get a reliable signal almost all the time saying that something is there. You won't get false negatives and you won't get false positives. And that is the kind of extreme sensitivity that you need for the wide adoption of these PCR approaches to detecting somatic mutations relevant to cancer. So I don't know if that answers your question, but hopefully it'll help a bit.
Thank you again, Dr. Fred Kramer.